Good morning, everyone.
Good morning.
Morning, ma'am.
We'll start with the classes now. So I'm going to share my screen here. Okay, so today's topic is translation. Okay, so last class we have discussed about the transcription process, that is synthesis of mRNA. So the next step in the protein synthesis is the translation. Okay, so this transcription and translation together is called as the gene expression. Okay, so once the mRNA is synthesized, the mRNA after the post transcriptional modification, mRNA is transported to cytoplasm for the synthesis of protein. Okay, so that synthesis of protein is called as translation. Okay, so we'll start with the steps of translation. So translation, as you know, the genetic information which is stored in the DNA is getting transferred to RNA, that is mRNA, during the process of transcription, and ultimately this mRNA is getting expressed in the form of protein. Okay, so biosynthesis of protein is called as the translation. Okay, so protein is made up of amino acids, and this amino acid, the chain of amino acid, okay, so the polypeptide chain which is getting synthesized is called as the translation. In the sequence of amino acids, so what which amino acid has to be added to the growing polypeptide chain, that is based on the codon sequences in mRNA. mRNA, so we know codon is what? Codon is the set of 3 nucleotides, isn't it? Okay, so this set of 3 nucleotides determine which amino acid has to be added to the growing polypeptide chain. So whatever the codon sequences which is present in the mRNA, based on that the amino acid will be carried by the tRNA, and the carried amino acid is getting added to the growing polypeptide chain. Okay, so based on the what amino acid are added to the growing polypeptide chain, the protein will be synthesized. Okay, so here, so from this it is very clear that which protein has to be synthesized, that is based on the codon sequences in mRNA. Okay, so the mRNA gets codon sequences from where? mRNA is getting the codon sequences from DNA. Okay, so then the information which is stored in the DNA, so whatever the information, how it is stored, how the nucleotide sequences are present in the DNA, based on that the proteins will be synthesized. Okay, so that is why we say that the genetic information which is stored in the DNA is getting expressed in the form of protein, because so based on the codon sequences in the DNA, mRNA is synthesized. So based on the codon sequences in the mRNA, amino acids will be added. So how in what order, in what series the amino acid is getting added to the polypeptide chain, based on that the protein will be synthesized. Okay, so that means the protein, which protein has to be synthesized, that information lies in the DNA. Is it clear to you?
Yes, ma'am.
Yes, ma'am.
Next is coming to the biosynthesis of protein. What are the stages or the steps involved in the protein synthesis? That is generally there are 5 main steps involved in the protein synthesis. The first one is requirement of the components. Okay, so what are the basic requirements for the protein synthesis? Like all these 5 steps we'll be discussing individually. So the first step is the requirements of the components of biosynthesis of protein, that is like different mRNAs, amino acids, then certain protein factors, then energy components. So these these are the basic requirements for the protein to get synthesized. Then the next step is activation of amino acid. That is, if all the required components for the protein synthesis is ready, then the amino acid is one of the main requirement for the synthesis of protein synthesis. Then once the requirements are ready, the amino acid has to be activated. Okay, so the second step is the activation of amino acid. Okay, so it requires some enzymes and the energy molecule. Then the third step is the synthesis of protein. Okay, so which is again divided into 3 different stages, that is initiation, elongation and termination. Then the fourth step is the chaperones and the protein folding, and the fifth one is post translational modification. This chaperones and protein folding can also be categorized under post translational modification, because immediately after the third step, that is protein synthesis, after that the protein which is synthesized here is an inactive molecule, okay, which has to be converted into the active form of the protein. So that modification is categorized under the post translation modification. The chaperones and the protein folding is also a part of that actually. So this fourth and the fifth step, it includes the post translational modification. Okay, so modification by which the inactive form of the protein can be converted into an active form. Okay, so so these are the 5 steps involved in the synthesis, that is translation process. We'll see one by one. That is, the first one is the requirement of the component. Okay, so what are the essential requirements for the synthesis of protein? So the protein synthesis, we know transcription is happening in the nucleus, whereas translation is happening in the cytoplasm. Okay, so the site of protein synthesis is mainly ribosomes. So ribosomes are the place where the proteins are getting synthesized. Okay, what is the first requirement for the protein synthesis? That is the amino acids. Amino acids, the polypeptide chain is made up of amino acid. Okay, so proteins are the polymers of amino acids. Okay, so different amino acids joined together to make the protein. Okay, so the protein synthesis will take place only if all the essential amino acids are available. Okay, so all the required amino acid has to be available for the protein synthesis. Okay, so any kind of deficiency of the amino acids will definitely affect the protein synthesis. Okay, so all the required amino acids must be present for the protein to get synthesized. So amino acid is one of the main requirement of the protein synthesis. The next component required for the protein synthesis is the ribosomes. Okay, so why the ribosome? Because ribosomes are the site of protein synthesis. Okay, so it is the place where the protein synthesis is happening. Okay, so ribosomes, as we know, so ribosomes are present in the cytoplasm, rough endoplasmic reticulum. Okay, so mainly on the surface of the roughness of the endoplasmic reticulum is because of the presence of ribosomes. Okay, so ribosomes are present on the surface of endoplasmic reticulum. Here the ribosomes, when we see the structure of ribosomes in prokaryotes as well as the eukaryotes, ribosomes consist of 2 subunits. So I hope you remember the shape of the ribosome. So here it consists of 2 subunits. The one is big subunit, the other one is smaller subunit. And also we have several times here discussed about the sites in the ribosomes. Ribosomes consist of mainly two to three number of sites, that is A site, B site, and also one more I have already explained, that is exit site. So here the A site is mainly is the place where the amino acyl tRNA comes and bind. So what is this amino acyl tRNA? Amino acyl tRNA is a tRNA which carries the amino acid. Okay, so in the different types of RNAs also we have discussed, that is tRNA, rRNA, and mRNA. So you remember the 3' end of the tRNA is the place where the amino acid binds. Okay, 5' end and the 3' end. 3' end, one particular codon is present in the 3' end, which is CCA. Hope you remember it. CCA is the place where amino acid is getting attached to the tRNA. Okay, so A site in the ribosome is a place where this amino acid tRNA, that is tRNA, which carries the amino acid, comes and binds to that to the RNA. Then P site is the second site where the peptidyl tRNA gets attached. Okay. We will see that in the elongation process of translation. Then in eukaryotes, there is one more site which is called as exit site. That is, it comes after A site and P site. Okay, so it is a place from where the polypeptide chain is gets removed from the complex of mRNA and the ribosomes. So that site is called as the exit site. Okay, so basically there are 3 sites in eukaryotes, that is amino A site, P site, and the exit site. So here I will show a picture of ribosomes, mRNA, and tRNA complex. You can see here. Okay, so this is the here is the ribosome. So here you can see this portion of the ribosome is called as the smaller subunit, and whereas this is the bigger subunit of the ribosomes. So here you can see the mRNA from 3' end to 5' end containing the nucleotides. Okay, so like a set of 3 nucleotides which are called as the codons. Okay, so here the mRNA is it is getting attached to the junction between the bigger subunit and the smaller subunit actually. So in this picture, even though it is showed in the smaller subunit, it is in the junction between the bigger subunit and the smaller subunit. Then here also you can see the 3 sites in the ribosome, that is A site, P site, and the exit site. All these 3 sites, that is A site, P site, and the exit site, it covers both bigger subunit as well as a smaller subunit. Okay, so you can see this A site covers both bigger subunit as well as the smaller subunit, P site also, and also the exit site. All 3 sites covers the bigger subunit as well as the smaller subunit. And here you can see this is the amino acyl tRNA. Okay, so the tRNA and the anticodon arm of the tRNA, which carries the complementary nucleotide. Okay, so whatever the codon which is occupied in the acceptor side or the A side, the complementary tRNA anticodon arm of the tRNA will have the complementary codon. Okay, and this tRNA, it carries the amino acid, and this amino acid is getting added to the growing polypeptide chain. Okay, so here this is the growing polypeptide chain, and this peptidyl tRNA which attaches to the P site. Okay, so here you can see the complete union of ribosomes, mRNA, as well as the tRNA. Okay, so this is the picture which shows the protein synthesis. That is attachment of mRNA, tRNA, as well as the polypeptide chain. Okay, so this is also another picture which shows the same components, that is ribosomes, bigger subunit and the smaller subunit, then mRNA and the 3 different sides of the ribosomes.
About exit site, ma'am. What's the use of exit site?
Exit site here. Exit, as I said, exit, as the name indicate, exit site is the place from where the once the protein synthesis is over, once the growth of the polypeptide. Here you can see this is one amino acid, this is another amino acid, this is the 3rd amino acid. Okay, so the whatever the 4th amino acid which is carried by the amino acid tRNA, it gets added to this growing polypeptide chain here. Okay, so once all the polypeptide, once all the amino acid is added, once all the polypeptide chain is completed, then only this component, that is the growing polypeptide chain with the peptidyl tRNA, comes to exit site, and once it comes to exit site, it gets removed out. Understood? Okay, so that exit site, this complete union, that is growing polypeptide chain and the tRNA, this tRNA will come to the exit site only when the protein synthesis is over.
Okay, ma'am.
Okay, so that we will see in the termination stage of protein synthesis. Okay, so now we are in the starting of the translation stage.
Okay, ma'am.
Okay. Okay, so the next component for the protein synthesis is the mRNA. So mRNA, we know, like which amino acid has to be added to the growing polypeptide chain. That information is stored in the mRNA. Okay, so because mRNA is transcribed from the DNA. Okay, so here the specific information which is required for the synthesis of a given protein is present on the mRNA. Okay, so that is specific information in the sense which protein has to be synthesized. So which protein has to be synthesized means the protein is dependent on the kind of amino acid is getting added to that. So amino acid, which amino acid should be brought by the tRNA is based on the codon sequence in the mRNA. Okay, so the information which is required for the synthesis of protein is present in the mRNA. So this information is passed on from the DNA. Okay, so DNA has passed on the genetic information in the form of codons to the mRNA to synthesize the protein. Okay, then the next component required for the, so far we have discussed about the first component is amino acid, next component is the ribosome, then third one is mRNA, and the fourth required component for the translation is tRNA. So tRNA, we know tRNA is nothing but it is the transfer RNA. So this transfer RNA, it carries the amino acid and hand them over to the growing peptide chain. Okay, so growing polypeptide chain. So it can transfer, the function of the tRNA is to carry the particular amino acid. So that amino acid was this tRNA which carries the amino acid is called as amino acyl tRNA. It comes and attaches to the acceptor site of the ribosome, and then this amino acid is handed over to the growing polypeptide chain. Okay, so that is once the amino acid is handed over to the growing polypeptide chain, amino acyl tRNA will be, this tRNA will be recycled back. Okay, so it will go out and will carry the next amino acid. Okay, so it is a cyclic process actually. Okay, so then okay, so tRNA, the transfer RNA carries the amino acid to the ribosomes. Then amino acid is covalently bound to tRNA at the 3' end. Okay, so tRNA, when we discussed about the structure of the tRNA itself, we discussed that is 3' end of the tRNA, which is like slightly longer end. Okay, so in the right hand side. So here that end of the tRNA, it carries a particular sequence. For all the tRNA, 3' end consists of CCA nucleotide. Okay, so this CCA nucleotide, that the last one is the adenine, cytosine, and adenine. This to this adenine only the new amino acids are getting added. Okay, so here to the nucleotides of the last nucleotides of the tRNA, the amino acids are getting covalently bound. Each tRNA has 3 nucleotide base sequences, that is anticodon arm. Okay, so anticodon arm, what is this anticodon arm? You remember the down portion of the tRNA consists of 3 set of codon, which is complementary to the codon in mRNA. So this that this complementary codon can base pair with the codon of mRNA. So that is why these codons are complementary. They are called as the anticodon because they are complementary to the codon in mRNA. Is that clear?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
So next we are going to the energy requirements. So what are the energy molecules which are used in the process of translation? So generally both adenosine triphosphate, that is ATP and GTP, okay, so are required for the supply of energy, because in many places we'll see this process of translation, it is an energy dependent process, okay, so it requires energy, because for the activation of the proteins and the transfer of amino acids to the growing polypeptide chain, in all these different stages, large amount of energy is required. So generally ATP and GTP are the energy molecules which supplies the energy for the synthesis of protein. And then the next component which is very much essential for the protein synthesis is the protein factors, okay, so these protein factors it is actually in general it is called as protein factors, but based on the stages of protein biosynthesis they are categorized in different names. That is, if it is like during the factors which are required during the process of initiation is called as initiation factor, and those which are utilized during the elongation is called as elongation factor, and during the termination these factors are called as a termination factor, okay. So many initiation factors and elongation factors and termination factors work coordinately to conduct the process or to carry out the process of protein synthesis, okay. So availability of these protein factors are also very much essential for the synthesis of protein. Okay, so we'll see the what is the role of these factors, that is initiation factor, elongation factors, and termination factor when we discuss about the initiation, elongation, and termination. Next is about the next step, that is, so far we were discussing about the requirement of the components. What are the essential requirements for the protein synthesis to happen? That is amino acids, then different types of RNAs, then protein factors, and the energy requirements, okay. Then the next coming to the second step in the protein biosynthesis is the activation of amino acids, okay. So here the amino acids, we know the amino acids are carried by the tRNA. So for the amino acids to get attached to the tRNA and then transfer this amino acids to the growing polypeptide chain, it requires the amino acid has to be activated, okay. So this activation process of amino acid requires energy also. So with the utilization of energy and with the utilization of certain enzymes, amino acids are activated, and this activated amino acid is getting attached to the tRNA, okay. So then this tRNA, once the activated amino acid is attached to the tRNA, then this tRNA is called as the amino acyl tRNA, okay. So the amino acid has to be activated to get attached to the tRNA, as well as to transfer this transfer this amino acid to the growing polypeptide chain, okay. So how this amino acid is activated? So that is, here a group of enzymes which is called as amino acyl tRNA synthetase, okay. So the enzyme which is required here for the activation of amino acid is called as the amino acyl tRNA synthetase enzyme, is essential for this step. That is, this enzyme and this enzyme, it is highly specific for the type of amino acid and tRNA. Okay, so that means if the amino acid is, if it is a tryptophan, okay, so then the enzyme is also specific for the tryptophan, like that, okay. So if the amino acid is aspartic acid, okay, so the enzyme is also specific for that, okay. So not all, it is not a common enzyme, but it is the enzyme is specific for the tRNA as well as the amino acid. Okay, so that class of enzyme is called as a amino acyl tRNA synthetase enzyme. Okay, so these enzymes, it gets amino acid first get attached to the enzyme with the utilization of energy molecule. Okay, so first that step is actually called as the activation of amino acid. So here, by utilizing one ATP molecule, the amino acid gets bind to the enzyme, that is to the synthetase enzyme, and it forms a complex called as enzyme, then AMP plus AMP, that is with the removal of 2-phosphate molecule, ATP is getting converted to AMP. Okay. So enzyme, AMP, and amino acid complex. Okay, so when the amino acid is in complex with the enzyme and the AMP molecule, the amino acid is activated. Okay, so that is the union of enzyme, then AMP, then the amino acid. Okay, so that complexation is called as the activation of amino acid. Okay, so here amino acid is transferred. Once this complex is formed, the complex of amino acid plus enzyme is formed, then this complex is transferred to the 3' end of the tRNA, and once this amino acid is transferred to the tRNA, then the tRNA is called as the amino acyl tRNA. Okay, so here you can see that here you can see that the first step you see here, this is the enzyme, that is amino acyl tRNA enzyme, plus the amino acid and the utilization of energy molecule. Here you can see this, it gets with the utilization of the energy, that is here you can see 2-phosphate molecule is removed, 2 phosphates are removed here, you can see here. So then it forms the enzyme, amino acid, and the AMP complex is formed. Okay, so once the amino acid is attached to this complex, once the complex is formed, amino acid is activated. Okay, then this activated amino acid will be then this enzyme mediates the attachment of this activated amino acid to the transfer RNA. Okay, so this is the tRNA. You can see this this end of the tRNA is the 3' end, which has the specific the CCA codon, that is you can see cytosine. You can see here cytosine and the adenine. Okay, so this is the CCA end of the tRNA. To this CCA end of the tRNA, the amino acyl tRNA transfer the amino acid. Okay, so here you can see this is the amino acid which is transferred to the CCA end of the tRNA. This is the tRNA. This is the 3' end of the tRNA, which has cytosine and adenine nucleotide. To this adenine nucleotide, the amino acid is being transferred here. Okay, so here there are two class of synthetase enzyme, that is class 1 and class 2. So based on the class of based on the type of the synthetase enzyme, this amino acid is attached either to the 2nd position or to the 3rd position. Okay, so however it is either to the 2nd position, if it is a class 1 synthetase enzyme, then the amino acid is transferred to the 2nd position, and if it is a class 2 enzyme, the amino acid is transferred to the it is just however it is, the amino acid is getting attached to the 3' end, there is a small variation to which place it attached, that is based on the type of the synthetase enzyme. Class 1 brings the amino acid to the 2nd position, and class 2 brings the amino acid to the 3rd position. Okay, once the amino acid is attached to the tRNA, this synthetase enzyme and the AMP will be recycled. Okay, so that will be that get detached from the amino acid and will be utilized for the activation of next amino acid. Okay, so this is getting, once this amino acid is transferred to the tRNA, the tRNA becomes the amino acyl tRNA. Then this synthetase enzyme, then the AMP will be recycled back. Okay, so will be utilized for the next activation of another amino acid. Okay, so this step, this entire step is called as the activation of amino acid and the formation of amino acid tRNA. Okay, so this is how amino acid is getting attached to the tRNA. Amino acid simply cannot come and bind with the tRNA. It requires to get it, the amino acid has to be activated. So only the activated amino acid can get attached to the tRNA. Okay, so this activation of the amino acid is done with the help of amino acid tRNA synthetase enzyme. Is it clear to you?
Yes, ma'am.
Yes, ma'am.
Okay, so this step is called as the activation of amino acid. Okay, so I stop here. We will continue it in the next class. Okay, so thank you all of you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.